I think there are a lot of considerations, but the three limitations or three considerations that I'm going to focus on, not to say that this is an exhaustive list. Number one, how do we ensure that the genes that we are editing, we are only editing them in the cancer cells? That we are not editing them like if we were to give a person these CRISPR-Cas, how do we make sure that we're editing these genes in the cancer cells and not editing these genes in the normal cells? Because if we edit the genes in the normal cells, it may have detrimental effect, right? I mean, these are genes that we're trying to turn off or on that control the cell behavior, that control division, etc., the cells growing, dividing, surviving. So how do we make sure that we are only impacting the cancer cells and not impacting the quote unquote normal cells and altering those genes in there? Second, how do we deliver these CRISPR-Cas to the tumors? Like a delivery mechanism, there is a lot of discussion about using viruses to deliver them or direct injection or so on and so forth. But delivering these like delivering them in an animal is easy, but delivering them in a patient much more complex. Especially that tumors also have altered blood flow, etc. And tying to the delivery question is the antigenicity of the Cas, because the Cas system comes from bacterial proteins, and there's a risk of developing antibodies against it if you are going to use it in repeated dosings in patients. So the delivery aspect, so the specificity, the delivery. And then third big question is ethical question. Like if we start altering these genes, how do we know that people are not going to use this to alter other genes that impact, let's say, things that are like related to our identity, right? Like things related to racial makeup or things related to certain disease types, like trying to target people who have like certain disease types, etc. So always, anytime you're altering the genome with any technology, there is a huge ethical consideration because really the genome is the blueprint of who we are, right? And developing that technology to alter the genome always has to have some ethical boundaries to prevent people from using it for altering other aspects of our identity. So those would be the three main things, right? Uh, how do you deliver it selectively and not into normal tissue? How do you actually assure that it can be delivered and prevent development of antigenicity and so on and so forth? And third, the ethical considerations of actually altering these genes in a human being and how would that impact other aspects of our identity? So epigenetic editing is a technology where we're targeting the areas of the genome that have epigenetic modifications, while the traditional CRISPR-Cas9, you are targeting the genome where you have a specific genetic sequence, like you're targeting a certain gene, right? Here, you're targeting areas of the genome where you have epigenetic changes. Of course, epigenetic editing would be very useful in tumors where epigenetic modifications are the driver of the disease, sort of like that colon cancer type that we talked about that happens in the right side of the colon, sometimes called CIMP, C-I-M-P. For example, for that type of a tumor where epigenetic modifications are a major driver, using epigenetic editing techniques would be maybe a better approach. And other tumor types where maybe it's a mutation that's driving the tumor, then going after that mutation with a CRISPR approach would be better than epigenetic editing approach. So I think fundamentally would tie to the underlying mechanism that's driving the cancer and causing the cancer to behave in the way it is. So there have been a lot of there's other ways to target epigenetic modifications besides CRISPR and epigenetic editing. And those are to target the enzymes that are involved either in the epigenetic modifications, like adding the methylation to the genome, or targeting the genes that are involved in reading the areas that are modified, or targeting the genes that are, because we have genes also that demethylate or deacetylate, so that erase the effect of epigenetic changes. So targeting those three approaches has been clinically a very active area. There are drugs that are in clinical practice, not in colon cancer, but in other cancers that target epigenetic modifications. Target, for example, there are demethylating drugs that are in clinical practice. There are histone deacetylators that are in clinical practice, not necessarily in colon cancer. I think there are a number that are being studied in colon cancer. One class of drugs targets something called BET, and this is one of the enzymes involved in reading these areas that are epigenetically modified. That is an area of active research that I think holds promise in colon cancer. Again, the demethylating approaches have been tried in colon cancer, have not had really a great success up until now. But there are always more potent inhibitors that can come along, or inhibitors that have maybe select the patients better in future trials. There could be other venues for us to develop these drugs in the future in colon cancer.